Hello students, this is Kanika. Welcome to my channel PrepBiology. So today in this lecture we are going to discuss top 10 previous year's questions from chapter number 3 of your biology, that is the reproductive health. So the weightage of this chapter is of 5 marks and this chapter is very important from your unit 1. Let's start the lecture. So the first PYQ which have asked in your 2024 recent paper and 2023 previous year class 12th board exam, that amniocentesis is very useful and important techniques, but due to some reason there is a ban on amniocentesis. Justify this statement. So first of all, you have to know that what is amniocentesis. Amniocentesis, it is an important prenatal diagnostic technique. Amniocentesis is a technique used to determine any chromosomal, biochemical, enzymatic abnormality in the unborn fetus. So this is the technique by which we can diagnose any abnormality in the unborn fetus. So what happens is, in amniocentesis, it is very useful and important technique. So by which we can diagnose the abnormality in the fetus and we can treat the abnormality by diagnose it. So but it is banned. What is the reason behind it? So what happens is, amniocentesis is also used for the sex determination. So what happens is, in amniocentesis, the amniotic fluid is present in the placenta. So amniotic fluid is taken and the further tests of that fluid has been done by the in the laboratory. So what happens is, why it is banned? Because as this technique is used to determine the sex of the unborn baby, so some people misused this technique and kill the unborn normal female fetus. Female fetus, that is called the female feticide. So that's why this amniocentesis is legally banned, to avoid female feticide. So this is the answer of this question. Next question is, name the 2 copper releasing intrauterine device, that is IUDs. Intrauterine devices. So we have done this topic in the previous classes, that what are IUDs. So IUDs are the intrauterine devices. State 2 reasons that make them effective and contraceptives. So IUDs, the name copper releasing IUDs, we know copper T, copper 7, multi-load 375. These are the some of the copper releasing IUDs. State their reasons. So why they are effective? Because for human females, as the copper ion, they release the copper ion and suppress the sperm motility and fertilizing capacity of the sperm so that's why it is more effective contraceptives. Next, B part of this question is, how subcutaneous implants help in contraception. So we have studied that there are various subcutaneous implants which are also called as injections or implants or subcutaneous implants. So what happens is, how they are helpful in the contraception. So what happens, these are the small rod like structures, small rods which are made up of silicone, rubber, air, and they are flexibly surgically placed under the skin of the woman's upper arm. Woman's upper arm. So in these rods, they contain some capsules filled with the hormones, capsules filled with the hormones which helps in the contraception process. So these subcutaneous norplants or implants are helpful in the contraception. Okay, so this is the answer. Next question is, explain the mode of action of the contraceptive pills taken by the human females. Mention the schedule to be followed for effective outcomes. This is very, very important topic, that is the contraceptive pills. Under this, we have studied the Saheli pill mainly. Okay, so Saheli is the pill, contraceptive pills. These are the small doses of either progesterone or progestogen estrogen combination is another contraceptive method used by the females. Okay, so these pills contain the progestogen estrogen combination hormones and helps in the contraception in the human female. So mention the schedule to be followed for the effective outcome. So for the pills have to be taken daily. These pills have to be taken daily for a period of 21 days, starting preferably within the first 5 days of menstrual cycle. Okay, menstrual cycle. So then they are most effective. Okay, so this is the schedule to be followed for the effective outcome. They are taken daily, about 21 days, and preferably within the first 5 days, starting within the first 5 days of the menstrual cycle. So after a gap of 7 days of the menstruation, it has to be repeated the same pattern till the female desires to prevent the contraception. So this is the mode how they are taken. Okay. Next question is, name and explain the surgical contraceptives method that can be adopted by male partner. We know that under the surgical contraceptives, we have studied the vasectomy adopted by the male partner. Okay, vasectomy adopted by the male partner. It blocks. What happens is in the vasectomy, the vas deferens is tied and cut. So by this, we block the gamete transport and prevent the contraception. Okay, so it is the surgical method. The operation is done and the vas deferens is tied, cut, and it blocks. It is the permanent method of the birth control. Okay, the small part of the vasectomy is removed and tied up through a small cut on the scrotum to prevent the passage of sperm. Prevent the passage of sperm. Okay, so this is used by the male partner. If we talk about the female partner, the tubectomy is there. Tubectomy, this means the fallopian tube sides and cuts so that the eggs are released from the ovum, do not fertilize with the sperm. Okay, so this is tubectomy is used by the female partner. The difference between the tubectomy and vasectomy is very, very important. Okay. Next question is, explain the mode of action of copper releasing IUDs as a good contraceptive. So we know that the copper releasing IUDs are intrauterine devices are the copper T, copper 7, multilode C375. Okay, and we know that they release the copper ion. Okay, and they suppress the sperm motility and fertilizing capacity of the sperm. But how is hormone releasing IUDs different from it? So what is the difference between copper releasing IUDs and hormone releasing IUDs? The hormone releasing IUDs like LNG20, Progestasert, these are the hormone releasing IUDs. These IUDs make the uterus unsuitable for implantation. Okay, make the uterus unsuitable for implantation. So that is the difference between the hormone releasing IUDs and copper releasing IUDs. Okay, so you can see here that the previous year in your previous year's exams of the board, that IUDs have repeatedly asked, so make sure this topic is clear to you all. Okay, next question is, write the basic step followed in the ART program to help the childless couples. Okay, so what are the ART programs? These are the assisted reproductive techniques in which we have studied these different techniques like IVF, in vitro fertilization, ZIFT, that is the zygote intrafallopian transfer, GIFT, that is gamete intrafallopian transfer, ICSI, that is intracytoplasmic sperm injection, and artificial insemination, that is also called the IUI. So under the assisted reproductive techniques, what are the basic steps followed in the program of the ART? So if we talk about these all techniques and we talk about the basic technique steps followed in the ART program, so we know that first in the ART technique, assisted reproductive technique, the collection of gametes is there. First step is collection of gametes, that from male partner and female partner the gametes are collected. Next step is fertilization takes place. Fertilization takes place in the laboratory condition, in the lab condition, in the artificial condition, okay, or in the test tube. And the last stage is when the zygote is prepared in the artificial condition, in the laboratory condition, then the embryo is transferred, okay, and the embryo is transferred into the female reproductive system, that is the uterus. So this is the these are the basic steps that followed in the ART programs. Why is it known as a test tube baby program? So why it is named as the test tube baby program? Because ART technique mainly follow the IVF, that is the in vitro fertilization, that is also called the test tube program. Test tube program. Why? Because as the fertilization is carried outside the body, this is the test tube, and the fertilization is carried, the zygote is prepared in the test tube, the fertilization takes place in the test tube, that is the outside the body, in almost similar condition as in the body. That's why this is known as the test tube program. Okay, so this is the answer. The IVF technique is also very, very important. Next question is, why is in vitro fertilization, that is IVF, is so named? That I have explained. Because why it is? Because the fertilization takes place outside the body under the artificial condition. That's why it is called the in vitro fertilization. State its importance. So what is the importance of IVF? So IVF is an important technique because it helps to overcome the infertility. Okay, why? Because if the couple is unable to produce a child, then for them it is a very, very beneficial technique. By the in vitro fertilization they can produce a child. Okay, so this is the answer. Okay, next is distinct between the GIFT and ZIFT. So GIFT and ZIFT, GIFT is gamete intrafallopian transfer, ZIFT is zygote intrafallopian transfer. Okay, this is the main difference between the ART techniques. Okay, so in the gamete intrafallopian transfer is when the egg cells are removed from the woman's ovary and placed in one of the fallopian tube using the laparoscopy along with the man's sperm to be fertilized. So in this, the gamete is transferred directly. So in ZIFT, the zygote is transferred directly into the fallopian tube by the laparoscopy. So what is laparoscopy? Laparoscopy is a technique by which this is the needle-like structure is injected into the fallopian tube of the female, and the gamete or the zygote is transferred into the fallopian tube for the further development. Okay. So this is the basic difference. Read it carefully. Next question is, explain the following techniques: ICSI, IUI, surrogacy. So what is ICSI? ICSI is intracytoplasmic sperm injection. Okay. So as the name suggests, intracytoplasmic sperm injection. So the single sperm is injected directly through the help of the needle. Single sperm is injected directly into the cytoplasm of the ovum. So this is the sperm and this is the ovum. Okay. So in ICSI technique, the intracytoplasmic sperm injection technique, the single sperm is directly injected into the cytoplasm of the ovum. What is IUI? IUI is intrauterine. IUI is also known as artificial insemination. IUI is known as the intrauterine insemination. Intrauterine insemination. Okay. So what happens is, it is also called the AI, that is artificial insemination. What happens is, the collection of the semen is collected from the donor or the husband, and it is artificially introduced directly into the vagina of the ovum, that is called vagina of the female. That process is called the IUI, that is intrauterine insemination. That the sperms from the donor father, okay, directly injected through the needle, directly injected to the vagina of the female, okay, for the fertilization. Okay. So next is the surrogacy. What is surrogacy? How it is important? Surrogacy is the use of the gestational carrier, okay, use of the gestational carrier or the another woman. It is the another woman that carry the embryo, or the donor embryo to term. Okay. So what happens is, the embryo is this is the couple, that is intended couple, which is unable to fertile, okay, so which is unable to produce a child. So what happens is, the male and female gametes are taken from this couple, and IVF technique used, and the zygote is prepared in the laboratory condition. Then after the development of the embryo, this embryo is transferred in the another woman, that is the gestational carrier, and that carry the embryo for about 9 months up to the delivery. And after the delivery of a baby, this baby or this child is given to this couple. Okay, so this is called the surrogate. Question. This is very, very important and different question that have asked in your 2023-2024 board exams. The A part of is this: as the part of ART, that is assisted reproductive techniques, what is the destination for blastomere with a count of less than 8 cells or more than 8 cells? So we know that in ZIFT, that is zygote intrafallopian transfer, the 8-celled blastomere is preferred. Transfer of the 8-cell blastomere is preferred. Okay, when the embryo getting 8-celled blastomere, then that embryo is preferred for the transfer, okay, into the fallopian tube. 8-celled blastomere is preferred. So what is the destination for blastomere with the count of less than 8 celled blastomere or more than 8-celled blastomere? So you have to answer this question. So for the less than 8-celled blastomere, it is directly transferred. It is directly transferred into the fallopian tube. Okay, into the fallopian tube of the female. But if we talk about more than 8-celled embryonal state, it is transferred to the uterus. Okay, so this is the difference. Next question is, what could be the reason behind transferring to the destination identified in A? So what is the reason behind it? So the embryo with more than 8-celled blastomere are generally considered healthier. Okay, so they are considered healthier. Why? Because they have a better chance of successful implantation, for the successful implantation. Okay, that implantation. So for the successful pregnancy, the more than 8-cell stage is more preferred. Okay, for the further development, hence they are placed in the uterus, not in that fallopian tube. C part is, what technique are used to transfer the blastomere to destination identified in A? So what techniques are used for less than 8-celled stage? That is ZIFT is used, that is zygote intrafallopian tube is used. But more than 8 blastomere stage, we use the IUT, that is intrauterine transfer. Okay, so these are the two techniques which are used for less than 8-celled blastomere transfer. We use the ZIFT, that is ZIFT. For more than 8 cells transfer, we use the IUT, that is intrauterine transfer. So this question is very, very important. Okay, the last question of this lecture is, a non-government organization, that is NGO, aim to increase awareness against STD. Okay, so this topic, STD, sexual transmitted diseases, or STIs, that is sexual transmitted infections, are very, very important. A part of it is, what could be the ideal target age group for the NGO? For the non-government organization, they help to increase the awareness against the STDs. Okay, so what could be the ideal target age group? The ideal target age group for them is 15 to 24 years. 15 to 24 years adults. Why? Because in this particular age, the hormonal changes is maximum. So maximum chance of STDs are there. Next is B part, mention any two potential long-term health-related complications of untreated STDs that the NGO should educate the target age group about. So for there are, mention any two potential long-term health-related complications of untreated STDs. So first one is the chronic pain and discomfort in various body organs. Chronic pain and discomfort in body organs. Okay. Next will be the infertility. Infertility in some STDs. Some STDs is seen. Okay. Third is lower immunity level and increased risk of other infection. Lower immunity level. Immune system is lower. And the last one is the high risk of transmitting the disease to others. High risk of transmitting the disease to others. Okay, to others. So these are some long-term health-related complications and of the untreated STDs. So the NGO should educate the target age group about. Okay, so this is all about the today's lecture. So if you want to check out some STDs which are in your syllabus, sometimes the MCQs have asked in your previous years related to STDs or STIs, sexual transmitted diseases or devices. So you can just revise this chart of STDs, that is syphilis, gonorrhea, AIDS, hepatitis B, genital herpes, genital warts, chlamydiasis, trichomoniasis, okay, candidiasis. So these all are the sexual transmitted infections or diseases, and the causal organisms are also important. So please check out this chart and revise this chart. If you like this lecture, like, share and subscribe my channel, that is Prep Biology. Okay, so thank you so much.